Welcome to this series on bioinformatics sponsored by CEC Gurukul lectures. We've been talking about several aspects of bioinformatics till now we have talked about, and we also talked about the metagenomic data analysis. I introduced you in the earlier section about this metagenomes or microbiomes, what they are and what they refer to. And in this one today, I intend to take this to the next level, meaning I want to give you practical exercise. How do you analyze metagenomic data if you are given a colon data? But before we go for that, I think it makes sense to first quickly revise the basic concepts so that our analysis is not stuck anywhere in between. So metagenome or metagenomics, as all of us know, is the study of microbes in their natural living environment, and this involves studying them as complex communities in their natural environment, because otherwise this has been very difficult to study them in their natural environment and along with their interactions. And here we believe in one concept that is called as OTU, operational taxonomic unit, and we consider that as a proxy of a species. So we study microbe as a gene sequence or a genome, as a part of their genome, and we consider them as a proxy for species. So this metagenomic methods, we learned that they are specially useful for the study of microorganisms that are not easily cultured in the laboratory, or because of certain reasons their taxonomic identification is difficult. Like, to convince you, I have an example of arbuscular mycorrhizal fungi here. These arbuscular mycorrhizal fungi, if we try culturing them is not easy, and it's a very lengthy process and a tedious, labor intensive process. So if we can study, and we cannot study them in the natural environment because of these limitations and several other associated limitations. But with metagenomic method, it becomes easier to study organisms like this. And similarly, studying them in the community, the metagenomic methods give us a much clearer picture of the interactions in a community. So metagenomics basically is stemming from genomics, but we believe we clarified how it is different from genomics. So in genomics, what we used to do is, in the environment, say for my arbuscular mycorrhizal fungi, once again, what I'll do is I'll take them out from the environment where they are growing, say for example rhizosphere. I'll collect them from the rhizosphere, bring them in the laboratory and culture them, multiply them, and when I have many of good specimens, I will isolate the DNA, amplify it and sequence, and then I would do the complete sequence, and then we complete sequencing of the genome. I would say we're talking about genomics. But when I am not culturing them, I am taking out all DNA from one environment, again the rhizosphere. What I would do is I will isolate all DNA from the environment, DNA isolation. After that, I will amplify all this DNA, I will assign this taxonomically, and then I would know which organism, just on the basis of sequences we know which microbes are present, meaning we don't go for culturing. So it's a shorter method and can be very accurate, but in the present scenario it has its own limitations. But it is a quicker method, that it tells us about many more sequences, many more microorganisms which cannot be cultured, and they still can be detected in metagenomics because we can isolate their sequences. So they are obviously different from genomic methods, but they are a part of genomics. So what we do in metagenomics, I made a colon here, I show you intestine. Why? Because this metagenomic methods and studies begin from the intestine or the gut microflora, The studies began where we studied the gut microbes associated with this gut, and we call this as gut microbiome, meaning along with the gut, when we study these organisms, we say we're talking about gut microbiome. So this metagenomic studies of gut microbiome or rhizosphere, I said earlier, can be of two types. One is sequence based metagenomics and the other is functional metagenomics. So in sequence based, what happens is we isolate all these sequences, we characterize these sequences. These type of studies are more useful in taxon identification and the interaction studies, which all bacteria or virus or microbes are increasing or decreasing their interaction. There also these are very useful. But in functional genomics, we talk about the functions performed by these microorganisms in the environment. Like in sequence based metagenomics, I told you already, we analyze the sequences from the environmental samples and we assemble them in genomes, we identify genomes and we try to understand the function performed by them. But in functional genomics, we isolate DNA from microbial community to study the functions of the encoded protein. So these are the basic two difference, but today we'll be doing mainly the sequence based metagenomic study, data analysis, and in there we will limit ourselves to 16S rRNA sequences, not the whole genome sequences which we talked earlier. Like, I would still suggest you to go back to our part 1 and part 2 lectures on metagenomics. Still, I have revised most of the concepts so that today's study is not hampered. In the another term, very quick reference to this term because it might be asked anywhere, what is the difference between metagenetics and metagenomics? Like the genomics and genetics we talk about one gene, in genomics we talk about the complete genetic makeup of one organism. So in both these things, but we still talk about one organism. So genetics, we talk about one gene of one organism. In genomics, we talk about all the genomes of one organism. In metagenetics, we talk about one gene of all genome in one environment, one community. In metagenomics, we talk about all the genes in the environment in one community. So they are similar in some way, but they can be different also. But the only difference here is that we talk about environment when we talk about metagenomics or metagenetics. Again, a similar term which pops in very routinely, when you talk about genomics or any other sort of study, you would see this term written metatranscriptomics. Now, what is metatranscriptomics? Like the transcriptomics, where we talk about mRNA or the gene expression. Similarly, in metatranscriptomics, we talk about the science that studies gene expression of microbes in their natural environment. So we study about the expression of the genes, and it also allows to obtain a whole gene expression profiling of the complex microbial community. So what we do is, here we study all the genes in one environment and then we focus on their expression profiling so that we could talk about the interaction. This also is very interesting and this has huge application in present day environment. Like, I tell you, the metagenomics has been known to advance the knowledge. It's a modern science and it has been adding to present day. In fact, even in our clinical studies or disease diagnostics, these metagenomic methods have been applied. So once you understand the analysis of this data and how to take out meaning of this data, it is routinely practiced in All India Institute of Medical Sciences and other medical institutes. So how metagenomics has helped in human health? What they do is that we do the sequencing to characterize the microbial communities from different sites of the different individuals. So there was a human microbiome project, and what was done in that is that sequences from the gut microflora or maybe the fecal microflora or maybe the skin microflora, that was analyzed and characterized, and when it was studied in detail, it led to the application of diagnostics and evidence-based medicine and has established itself as a powerful tool to address many of the issues related to personalized medicine. Like genomics is helping in the personalized medicine, equally metagenomic methods have been helping, and you will be able to appreciate this concept very nicely when we reach to the end of this lecture. By the end of this lecture, when we have actually analyzed one metagenomic data. So this metagenomic data analysis in medicine especially, and this human microbiome project, sequencing project, like the genome sequencing project, the metagenomic sequencing project for human beings, what did it do? It had a promise, and it was a sensitive and a rapid method to diagnose infection by comparing genetic material found in the patient's sample to the databases of already known several parameters. We have databases for everything. So when we, for any patient, for any biopsy sample, if we take out the sequences and if we analyze those sequences, and if we compare them with already known sequences for the several databases, like to cite some examples, the microscopic human pathogens, we have a database of that, and we have a database for viral, fungal, parasitic organisms, and the database on antimicrobial substances we already have gene sequences associated with clinical phenotypes. And I tell you that what is happening even in COVID-19 studies, we are using these databases, and every time when we are detecting new mutant or new combination with the host, this study is a present day study which is finding applications side by side. So same thing is being done with COVID-19 virulence and strains identification. So this human microbiome project was, one phase of that is already over, and it had led to several huge help in the personalized medicine and diagnosis of several microbial disorders. So that was what I was telling you about human microbiome project. And when we talk about human microbiome, we are talking about the collection of microbes in the human body, in any patient maybe. And I told you already, this project is finished now. And what did it give you? It gave you several surprises after this project. One was that we realized that microbes contribute more genomes responsible for human survival than humans' own genes. So what does that mean? When we talk about the microbes on several occasions, anywhere you are asked, if you remove the microbes, nothing is going to survive in this world. Same way, microbes, when we analyze this microbiome in human body, we realize that we have more microbial gene contributing to the well-being of a human than its own genome. So it was, not worth considering human genes in a body, excluding the microbes. That was an important fact which came out from this microbiome project. And other thing it gave us was that the bacterial protein coding genes are 360 times more abundant than human genes. So if I analyze my gut microbiome, what I know is that we have more of bacterial protein coding genes in my body than my own genes. And same way, when we compared with previously sequenced microbial genomes and human genome, the metabolic reconstruction of gut microbiome told us that there is a significant enrichment of genes involved in several metabolic pathways, and the metabolism of xenobiotics, or maybe the foreign substances, glycans, amino acids. So the point was that it is not that the microbes, the human genes are working. They will not be able to do anything in absence of these microbes association or the microbial genes associated with them, and we got to consider them along with. So in any medicinal treatment for a human being, it is now well established that we must consider the associated microorganisms with them. And the quickest method of knowing these associated microorganisms is to analyze their microbiome. So another project which is going on nowadays is Earth Microbiome project. What we are doing here in this is we have huge number of collaborators, and they are trying to know the all the microbial life on this planet, and that is going to give you wonderful results, and maybe we'll be able to improve our nature earth once we understand what all microbes are working and how they are interacting with these in the atmosphere. See, the point of these two examples, why I gave you these two examples, I just wanted to convince you that it is a present day science, and the moment you do any research or any study on this, it reaches to the hospitals or the environment or the agriculture people side by side. It has found huge applications in all the arena of present day life and studies and research. So that was, I was talking about the background, and I wanted to tell you that we'll be studying the metagenome data and we'll take it to the end, and we will know how this study is so useful. Practically we will assign them a name. So let's start with the learning objective for today's lecture. That was the background. So what we'll be doing today is we'll be learning about the next generation sequencing and the data files we obtain in any metagenomic studies, meaning how come that next generation sequences has become so useful in the studies of microbiomes, and what sort of data files we get. Once we get the files, how do we do the quality check, and how do we create a OTU table with that, and how do we give a taxonomic assignment for that, and how do we build a phylogenetic tree with this. So by now we already have a very good background about the OTUs, taxonomic assignment. I already told you NCBI, BLAST and the other methods of taxonomic assignment. We have also talked about the phylogenetic tree building in our previous lectures. I would just suggest you to go back and revise whatever we have learned till now in this series on bioinformatics, and we'll be talking about these 5 things, and all these things we'll be doing with one selected software, that is QIIME. I introduced QIIME to you in the previous section also, but still I will take that in a bit more details here. Is you know, let's take them step by step so that you understand what is happening in your data and what does your data contain. So we take sample, and the sample, you know, when it is a gut microflora, it's a simple biopsy sample which has been taken from any patient. And what happens is, with this biopsy sample, what you will do is you will isolate the whole DNA, and we have very easy kits available for this DNA isolation, and then we amplify this DNA, and you know, we run the sample, but we not only isolate the DNA and try amplifying. We run 3 types of controls here. One is a negative control, where this sample DNA is absent. We do it without sample. We have a positive control where a known already identified sample is added, and because we see a lot of multidimensionality and variation in the data, it's an environmental data. And we also run side by side a mock community. What does that mean? That means we have already predefined combination of artificial or synthetic community, where we have predefined set of microorganisms present. So what we do is, if they are detected right by our data, our run of the experiment, meaning our things are going fine. So we run along with the sample DNA, we run 3 types of controls here. We have another very important thing which we must know is selection of markers or primers for PCR. And you know, in amplification, I told you already, high throughput sequencing has made it much easier. I'll talk about that little bit more in detail, that how that has made it so easy. And we have very important segment, that is primers. So that will come again and again in between, and we have talked about that in earlier lectures also, how primers are important, and deciding the primer makes it more important that whether you're going to get good result for your study or not. So coming to the next generation sequencing, you know, why next generation? We had several generations of these sequencing methods. The first generation sequencing, that was Maxim Gilbert method. We had second generation sequencing, which massively increased the throughput by parallelizing the many reactions. We had third generation, which allowed the direct sequencing, and after these 3 generations we reached to the next generation sequencing. And what happens in next generation sequencing is a massively parallel sequencing technology that offers ultra high throughput, scalability and speed, and the technology is used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA, and it is very useful in metagenomic studies. Why? Because it gives you a very accurate result, and the speed also is very good, and the costs also have come very low. So I have several examples of here, but the study which we are talking today is using Illumina sequencing because of the obvious choice of the good speed, low cost and the reasonably good rate of accuracy, because in the environmental samples to have a accuracy is very important. We must have the accurate determination of the basis, and you would realize that every step we are talking about the data quality or the quality check. So the advantages which we had with NGS and which made these studies possible was the ability to sequence hundreds of the sequence or gene regions simultaneously. It had higher sensitivity to detect the low frequency variants. It was very fast for high sample volumes. It had a detection limit which was much lower, and it allowed sample multiplexing. We'll talk about multiplexing a lot more in details, and it allowed the comprehensive genomic coverage. So I'll talk about multiplexing maybe just now, or here also I can tell you, but before that I like to tell you that the sequencing or the high throughput sequencing, the Illumina sequencing, gives us 4 types of files. One is the mapping file. The mapping file of our experiment will contain the metadata. What does that mean? That means it will contain the information data. In a way, it will tell you the hypothesis of your experiment. Like the experiment which we are studying is having the gut microbiome data. It has been taken from 2 individuals and it takes it from 3 sites of that individuals. So this all is given in the mapping file. That would you realize just now when we open up a mapping file. So mapping file, maybe you have to design a mapping file, and there you will be able to give all information. You must give, because the one who is studying your data after you should know your experiment, your hypothesis, and how you have done this. Another file which we get in this Illumina sequencing in metagenomic data is barcode file, and it contains the information to demultiplex these samples. So I come back to demultiplexing or multiplexing. So what is multiplexing is the samples, when we are amplifying them, in amplifying and sequencing we mix the samples. They are pulled together and they are sequenced simultaneously. So we have to keep a track of which sequence is coming from which sample. So I come back to this again. I said, what is multiplexing? Multiplexing is when the samples are pulled together. The samples are sequenced simultaneously in the Illumina instruments. This is multiplexing. And to keep track of our sample, meaning which sequence is coming from which sample, we assign them, and this assignment is called as demultiplexing. So first step in sequence assignment should be that we should know which sequence is coming from which sample. This is called as demultiplexing, and we have barcode files that will be able to tell you that. Like I just took one example here where I am telling you that how in amplifications the sequences are amplified and they are multiplexed, meaning all of them are mixed together. So at the first round itself, I have added 2 barcode sequences here. One is orange and the other one is purple. So in the very beginning, if I know my barcode sequence orange and purple, I would be able to identify that all these sequences belong to this sample. So this picture perfectly explains you that what is the multiplexing, that we mix them together after the initial start of assignment of this barcode sequence, and in demultiplexing we sort out which sequence belongs to which sample. The third type of file which we have here is the sequence file, which contains the sequences in FASTQ format. So FASTA everybody is very familiar by now, and the Q here is that these sequences, they also tell you about the quality of the bases in the sequence. So because we have huge data, huge variability, and it's an environmental data, to have a good information about the quality of the bases is also important. So for this metagenomic data, Illumina sequencing data, we have what? We have FASTQ format of the files. And the last type of file is reference data file, where we have already identified sequencing, meaning we have to have some already identified sequence to know the name of the sequence which we have identified for our sample or our data. So what we're going to do today is we'll install QIIME software from their website, we'll understand the type of files and the information contained in them, and we'll pick up the OTUs from there, we'll assign them taxonomically, we'll give them a name, and we'll try to build a phylogenetic tree with those sequences. So I said we selected QIIME for this one, and if you search, this is the most if you search this in Google, you will find it is the most popular software used around. Another one is Mothur. We talked about both of these in the previous lecture. Maybe you could go back and refer it from there, but here we are using the QIIME 2. The QIIME 2, the full name, if you want to know, is the Quantitative Insights into Microbial Ecology. And this philosophy of this software is it's a wrapper of best in class software, so we can do all these steps at one platform. That is the best thing here. It is extremely flexible, but it's not feature complete, and it is based on the language Python. It's open source, and University of California San Diego has developed it along with the North Arizona University. Those people, even now, they are very active, and they have very good community network to solve your problems whichever you want. So these are the several steps in metagenomic data analysis, which we'll be doing one by one in the next section. Thank you very much for your patient listening.